the last class we started looking at how the bacterial cells are grown for isotope labeling. So, the first step we saw is a transformation where we take the DNA of our interest, put it in the vector or plasmid, and that is inserted into the cell, and then we grow it on a agar solid plate, which is nothing but a petri dish, and in that dish we start seeing the colonies. This typically takes about 16-20 hours, let us say a day, and after a day you can come and see these colonies would have grown on your plate, provided you have selected for the correct marker, because remember ampicillin, if it is not resistant to, the contaminants also can grow. So, we need to have ampicillin resistance built into the cell. So, this is an important point, we need the ampicillin resistance built into the cell, which I said we can do with simple recombinant DNA approaches. So, now once these colonies have come, we can choose any of these colonies. You can see there are millions of them. We can choose any of this, and then what we do is we put it in a small medium. Again, this is 10 ml to 20 ml. You can see this is like a test tube here. So, in the test tube we can put that colonies and then grow it overnight at 37 degrees in the minimal medium. So, this is how it will become turbid after it has grown for overnight. Okay. So, this is a turbid, you can see this is a milky type of a solution it has become, because this is full of bacterial cells. Okay. So, again remember in this medium what are the sources of carbon and nitrogen? This is going to be a minimal medium for labeling, isotope labeling. In a minimal medium we have carbon 13 or carbon 12, glucose as a source of carbon, and ammonium chloride as a source of nitrogen, and other than these two we have to add some more components such as a salt or a buffer, it has to be buffered, and also you need magnesium chloride, magnesium sulfate, zinc and vitamins, but those are in very small quantities. The major component here we are talking about is carbon 13 or carbon 12 glucose and nitrogen, ammonium chloride. So, overnight this 10 to 20 ml culture is grown like this, and once it becomes turbid we transform it or we transfer it to a larger volume. So, you can say this is a medium which consists of again the same minimal medium. We take a larger volume around 250 or 500 ml, and this whole solution is just transported or transferred, or you can take the cells, you can centrifuge it, take the cells and put it in this medium. Okay. Now, this medium is again the defined minimal medium which contains the carbon 13 as a source of glucose as a source of carbon, and ammonium chloride and other materials, ammonium chloride mainly for nitrogen. So, this is very important that this is all throughout we are using minimal medium everywhere. Okay. Sometimes people use some other medium here at this stage and then transfer it to minimal, that is also okay. Now, after this transferring this liquid from here to here, we need to grow it again, this particular culture. So, this is grown in incubator shakers which look like this. This is a small incubator shaker, you can see the flask is here, and this is actually it will shake. Okay. So, this is rotated or shaken at a particular speed, that is called rotations per minute, RPM. Okay. So, that and the temperature inside is kept at 37 degrees, because that is the optimal temperature at which E. coli cells can grow. Now, the question is how long do you grow these cells? You grow the cells till the turbidity. So, this is this turbidity which looks like this is measured with a spectrophotometer. So, you take 1 ml of this, take it to a spectrophotometer. So, you need a UV visible spectrophotometer in your lab. So, you take that to that spectrometer, and at 600 nanometers, the wavelength is set to 600 nanometer, you measured what is the absorbance, or we use the word optical density. If that absorbance is around this value, then that is enough for the growth for the cells. We do not have to grow it more. Okay. So, this is the important point. The cell density, this is called the log phase, means the cells are very high in number, they are dividing very rapidly, and we have to now do the next procedure at this stage. So, you should not overgrow the cells. If you allow it to grow to 1.2, 1.3, then the cells will start dying. Similarly, if you grow very less 0.2 or 0.3, then the cells obviously would have not grown very enough. So, your protein will not come out in a good quantity. So, this is the stage to which the cells are grown, and as I said they are monitored using UV visible spectrophotometer by setting the wavelength to 600 nanometers, and in this wavelength you are measuring basically the scattering. Okay. So, this is not that it is absorbing this light, it is a scattering by this the turbid the cells, because the cells are also turbid. So, we are measuring the turbidity of the solution. So, once it reaches 0.8 to 1, what do we do next? The next step is called induction. So, this is shown here. So, now this is what we do now. So, what is induction? Induction is the transcription of the desired DNA, protein DNA, that is, remember we took the gene of our interest and we have put it in the plasmid. Now, the plasmid it is a DNA form. So, it has to be transcribed, or the transcription of that DNA to mRNA has to take place, because without that our protein will not be made, and that transcription begins or starts only when the inducers are added. What are these inducers? These are chemical inducers or physical, like for example, I can add a molecule known as IPTG. So, if I add IPTG to my solution when the density has reached this point, then that protein, that the RNA of that protein of our interest starts getting out in large numbers. Okay. So, the theory is that till we add these inducers, our protein is not so much expressed, means our protein is not expressed at all, it is blocked. But the moment we add these inducers, which we do not add in the beginning, we add only after some time when the cell density has reached appropriate value, and that point we add these inducers. These are chemicals, or it can be heat shock, I can do something else for induction, but typically in a standard very common approach is induction by IPTG, a molecule called IPTG. I do not have the molecular formula here, but you can look up in internet or any book. So, once I add IPTG to the cells, the cell starts now making the RNA of my protein of interest, okay, because the DNA which was there in the plasmid is converted now or transcribed to mRNA. And this is a step very important, because now this is at this stage is when our protein of interest is being made in the cell. Okay. So, that is done only at this appropriate stage. So, now what happens is you have C13 labeled glucose, let us say, in your medium, and my protein of interest needs C13 because it needs amino acids, it needs all the other things to make the protein. Now, all the amino acids have come from glucose, because glucose is the only source of carbon. So, if glucose is the only source of carbon, the amino acids are C13 labeled, and all the protein, the entire protein which is coming now from this DNA of my interest is also carbon 13 labeled. So, you see what we have done, essentially we have basically smuggled the DNA of our interest into the cell, and by smuggling we are basically fooling the bacteria, we are asking the bacteria to grow and make our protein in large quantity, although that is not its native protein. The protein which we are making is something foreign, means something alien to the cell. The cell naturally does not contain that protein, but we have smuggled the DNA of our interest into the cell, and we are making the bacteria make that protein for us in large quantities. And since the bacteria is growing on a C13 labeled medium, the entire protein is also carbon 13 labeled. Okay. So, that is how this labeling part comes into E. coli, through this kind of approach, in which the cells are grown in C13 labeled glucose, and the protein which comes out is also now C13 labeled, because we have made that DNA to express that protein in large quantity. So, this is the part known as induction. Now, once I add IPTG, I need to grow further. Grow means I need to keep the bacteria growing for some time. So, that step is not shown here, but the step after this, that after induction you have to still grow the bacteria for some time. So, I will write it here, you continue bacterial growth for, let us say, 4 to 6 hours. This is something which has to be optimized. Of course, there is no golden rule whether it should be 4 hours, 2 hours, 6 hours, 8 hours. It depends on the temperature in which you are growing. For example, let us say your induction happens at 30 degrees, then you need to grow a little longer. If the induction happens at 37, then typically 6-8 hours should be good enough. So, this has to be optimized for your protein if you want to get a very good yield. Yield means the output, this amount of protein. So, once that 4 to 6 hours is over, we have to, now our job is done, we have to centrifuge the cells and then break the cell. Lyse means break open. Why do we need to break open? Because remember, our protein which is coming from DNA to RNA is sitting in the cell, inside the cell. So, the cell has to be opened to remove our protein, but again if I open the cell, remember there are many more proteins in that cell, it is like a soup. So, the entire soup of the cytoplasm contains our protein as well as other proteins. So, we have to do something more later further which we will see, but first thing is we have to open the cell, break open the cell and take out all the cell proteins in the cytoplasm. That is provided our protein is actually in the cytoplasm and not in what is called as inclusion bodies. We will not worry about that. We will assume that our protein is soluble in the cytoplasm of the bacteria, and what we are basically doing is taking out the entire soup out of the cell. Now, this break opening is done typically with this instrument called as a sonicator, or there is another instrument called French press which we will not see. We will look at this sonicator. So, sonicator is basically something like this which has a probe which goes into the cell or solution of cells. We make a solution or what is called suspension. We take the centrifuge the cells and resuspend it in some solution, and then we break open the cell wall. So, essentially we are breaking the cell wall so that the entire cytoplasmic proteins come out. Once they come out, we have to purify our protein of interest, because remember, as I said, it is a soup, it contains many other proteins in the solution. So, we need to separate those proteins from our protein, and that typically is done by HPLC or FPLC. So, you need to use HPLC for some cases, sometimes FPLC is sufficient, and we can do that and get it in this form. Now, once we do that, the protein may be dilute, it may be not be very concentrated. So, if you recollect, you need a very high concentration of protein sample in NMR tube when we do NMR experiment. So, this is important to remember. We say that about 1 millimolar is required for a protein. Of course, for some proteins 1 millimolar may be too high a concentration, it may start precipitating and so on, but as high as possible we have to do, because NMR sensitivity very much depends on how much is the concentration. So, this is the final step where we take the purified protein coming from HPLC and we concentrate with some techniques like dialysis, or you can use Centricon and so on. There are different small instruments, and from based on that we can concentrate and put it in an NMR tube. After this you record NMR data. So, now this is a labeled sample now, and why do I say it is labeled? Because remember, right from beginning we have been growing the bacteria in a carbon 13 labeled medium or N15 labeled ammonium chloride. So, therefore, every carbon, every nitrogen of that protein of the cell is now carbon 13, and therefore, every protein is now carbon 13 labeled, not only our protein, also the other protein in the bacterial cell. But remember, we have separated those other proteins from our protein by using HPLC or FPLC or purifying our protein. So, what this NMR tube contains is purely our protein. So, the purity should be very high. Typically, the purity we can obtain is about 90 to 95%. So, 95% purity is good enough for NMR. If you get 99 and 100, that is even better, but typically that is the level of purity. And now, but every carbon in that sample should be C13 and N15, because we have labeled it. So, this is how a uniform labeling of proteins is carried out, uniform C13 and N15, because remember, we are not selectively choosing a particular amino acid type for labeling. We have chosen all the amino acids, everything is labeled with C13 and all N15 in case we want N15 also. So, now let us say this is what basically is the idea of uniform labeling, that is all the carbon and nitrogen in the protein are non-selectively labeled with C, means they are all uniformly labeled. So, this is what we said, you grow the bacteria. So, the question is how much glucose do we need? We typically for 1 liter of the bacterial culture we use 2 to 4 grams, 4 grams is on the higher side, you can also use 2 grams per liter, means a culture volume of liter. So, let me go back and show you what is it volume I am referring to. I am referring to this volume here. So, this is actually, the flask may be large, the flask may be 500 ml, but the culture is not 500 ml, this will be about 200 ml. So, if you have 1 liter of this volume in a big flask, for each liter of culture volume, you need to use 4 grams of glucose carbon 13 or 2 to 4 grams. For ammonium chloride the requirement is less, it is only 1 gram is enough for 1 liter of culture volume. So, these are the two things which we have to add, and of course, as I said, you have to also add salts and vitamins as a supplement. Okay. So, I would request you or recommend you to go to this paper and see more details of how we get this labeling done. Now, one more important thing is if you do uniform labeling. So, we are talking about uniform C13 N15 labeling, which means every carbon in the protein is C13 labeled. So, that means my neighboring carbon is also carbon coupling is present now in the molecule. Remember, for natural in the case of natural abundance, we ignored the carbon 13 coupling because because of 1% abundance, the chance that 2 neighboring carbons are C13 was very low. But in this case, we have now every carbon as C13. So, therefore, we need to worry about C13 decoupling. This is what is mentioned here, that once you have uniform labeling, you have to now use appropriate decoupling in all NMR experiments or pulse sequences, because if you do not do that, you will lose sensitivity by J coupling. The peaks will start splitting. So, you need to decouple carbon from carbon, carbon from nitrogen, nitrogen from hydrogen, and carbon from hydrogen. So, all possible decoupling have to be implemented or used to avoid any loss of sensitivity. This is something which is already built into the NMR experiments. So, like for example, when we discuss HNCA, HNCO-CA and so on, all those are already taken care in those experiments. So, in a standard NMR experiment if you are using, normally we do not practically we do not have to worry about this, because this is built into, it is default present in those sequences. So, let us move on now for a selective labeling concept. So, now what is selective labeling? So, selective labeling basically, as I mentioned, is selective labeling of a particular amino acid type. For example, let us say alanine or threonine. If I want to label only that amino acid, then only carbon, nitrogen in that amino acid is labeled with C13 and 15, and the entire remaining rest of the amino acids in the protein are now having carbon 12 or nitrogen 14. They are not labeled, isotopically labeled with C13 or N15. Okay. So, how do we achieve that? We will see shortly. But first, let us appreciate why do we actually need that? Why do we need to do this selective labeling? Because of the following reason. So, look at this schematic drawing here. This is a uniform N15 labeling protein, labeled protein, which means all the peaks, each peak corresponds to one amino acid here. So, this is something which we have seen in the first part of the course, how an HSQC gives rise to different peaks. So, each peak in HSQC corresponds to one amino acid. But let us say I have 3 types of amino acid in my protein. 3 types means, let us say one type alanine, B is something else, C could be something else. So, let us say only 3 types of amino acids are present in my protein. So, all the B's will come like this, all the A's will also show up, and all the C will come. But remember, these colors are only just to show you, but actually in a spectrum we cannot figure out which is A, which is B, which is C. It is just to illustrate this point I have shown in different colors, but in a real spectrum, NMR spectrum, in HSQC you will not be able to distinguish which amino acid type is each peak. They all come in this, they look all the same. But let us say now this looks little crowded here, and I want to get rid or I want to simplify the complexity of this spectrum. What I can do, I can only label A, I can choose to label A. If I do that, then only A will come in the spectrum, because only A will be labeled with N15 and all the B and C will not be, they will be unlabeled. That is what was mentioned here. B and C will be N14, but A will be N15. So, in that case only peaks corresponding to A will be present and amino acids B and C will contain N14, and therefore, they will not appear in this spectrum. So, by doing this I am actually, you can see here, I have reduced the complexity, I have reduced the number of peaks, I have made my life simpler. I can do the same with B now. I can do selective labeling of B and get another sample. Remember, each one is one separate sample. So, this sample only can now have A as labeled. I need to prepare another sample, go through the procedure which we saw in the previous slides and grow bacterial cells again, but now that cells will have only B labeled. So, that is another sample. I can make another sample where only C is labeled. So, like this for each amino acid I can prepare separate samples, but do we actually do that? Practically no, practically we do not do that way. We actually what we do is typically we choose a few type of amino acids which we are interested, which are for example very important for us. Alanine could be important, or lysine may be very important for the function of the protein. So, we choose a few amino acids which are in abundance, that is another issue. Suppose I have a lot of lysines in my protein and I want to selectively look at them, then I need to do selective lysine labeling. So, we do not, as I said, we do not really each take all the 20 amino acid and do one by one. That is not practically a good idea, because selective labeling is little expensive compared to uniform labeling. So, in uniform labeling like this, it is not so costly, but if I want to use selectively look at A or B or C, then I need to do something which requires money or cost, extra cost. And how do we achieve this? We will see shortly in the next slide. But remember, we have to choose a particular amino acid, a few amino acids, and label them selectively. So, let us see how the selective labeling is carried out. Again, this is very nicely explained in this paper. So, what we do is we take this, suppose we want to selectively label only alanine, but again remember I want alanine carbon 13 labeled. So, we take that amino acid, we purchase that amino acid commercially. So, it is available for example in many commercial companies. So, I can buy a carbon 13 labeled alanine for example. So, let us say I am looking at alanine, then how much alanine should I add? What I do is in the medium I will add this much per liter of culture, typically 100 to 200 milligrams. Okay. So, what we are doing is we are taking the medium, that's culture, cell culture. Remember the cell culture I showed you in the previous slides. We take the cell culture medium, and into that we add 100 mg of C13 labeled alanine, okay, and we add all other amino acids which are unlabeled. So, now what is happening? We are not adding glucose, we remove the glucose out of the medium. That means, I do not use glucose in the medium, but rather I am using each amino acid. So, I take 20 amino acids, but out of the 20 the 19 are unlabeled. So, they are added in unlabeled form, which is not very costly. One particular amino acid which I want to label is added in a labeled form. Okay. So, all of these are added in the same quantity, 100 milligram per liter of culture. So, now what does the bacteria do? The bacteria actually, now if you want N15 labeling, then in that case you take the N15 amino acid from the company, you buy this labeled amino acid from a company, and that is added in the same quantity, and other amino acids which you do not want to be labeled has to be in N14. For example, in our previous slide we wanted A to be labeled. So, if I go to the previous slide, this is what we want to be selectively N15 labeled, but B and C do not have to be labeled. So, in such cases, in this example we will take A in N15 form and B and C will be unlabeled form. So, we add those 3 to the culture, and the culture does not contain glucose, but rather it contains these amino acids. Now, what is the reason for that? The reason for that is if I add glucose, my carbon from glucose will start getting into the amino acid. So, that is something I do not want. I want the bacteria to only take those particular amino acids which are labeled. So, what the bacteria will do is that it will stop making its own amino acid, it will take the amino acid which you have given outside them in the medium. This is known as exogenous. So, this is known as exogenous amino acid. So, I am adding exogenously my amino acid, means I am adding from outside these amino acids, and what the bacteria does, it takes those exogenously added amino acid and takes it into its cell. It does not make its own amino acid. Why? Because you have given it for free in the medium. So, by doing that, it takes the amino acid which we have supplied, for example, alanine and all other amino acids, and starts using it for its protein preparation. So, whatever protein is made in the cell is now taken from these amino acids. So, this is basically how the protein will make the N15 label, because now the N15 label amino acid is actually incorporated into the cell. So, that means if I have my amino acid sequence like alanine, lysine, arginine and so on, my only one amino acid N15 will be this, and all the other amino acids will be N14, because I am supplying this in the unlabeled form and this is supplied in the labeled form. So, this is how the selective labeling of amino acids are done, and you can see that if I want to have both N15 C13 labeling, that means I want this entire alanine to have both carbon 13 labeled atoms and N15, then I need to do by this type of amino acid, means where C13 and N15 both are labeled in that amino acid. These are specially made amino acids which we have to buy from commercial agents, so from companies. So, once you buy those amino acids, you buy these also, but these are all unlabeled, but these are labeled, this is labeled. So, you mix all of this in this, each is 100 mg per liter of culture and put it in the medium. But remember, the medium does not contain glucose now, it is replaced by these amino acids, and what the bacteria does is simply one by one it takes all these amino acid into its cell. So, all these amino acids which are present in the medium is they all get into the cell, okay, and the bacteria now starts making all this protein using these amino acids. Okay. So, this is how the proteins are made, because now all the amino acids are available to it inside the cell. So, it takes these amino acids and makes our protein of interest, which is actually also made in the plasmid, which is sitting inside the cell, also gets now this labeled alanine, also gets this unlabeled lysine, also gets this unlabeled arginine and all those things. So, that is what is very important to know, and there is another thing which we will see in the next class, that this is not so simple as it looks here. Sometimes when I take alanine into my cell, it gets converted into another amino acid. For example, we saw in the previous few slides in the last class from a biochemistry perspective that amino acids interconvert, they convert from one amino acid to another. So, there is a danger that if I want to label lysine, it may also label some other amino acid, because inside the cell the bacteria converts one amino acid into another amino acid. So, these are the little difficulties which we have to deal with when we do selective labeling because of this interconversion. For example, let us say I am using glutamic acid. Glutamic acid or glutamine is basically known to transfer its N15 atom to all other amino acids. So, all amino acids actually get their N15 or N nitrogen from glutamine, that is called transaminase. We will see that in the next class and see how we can avoid such scrambling. We use the word scrambling of amino acids, and that we will see how we can deal with and avoid for improved efficiency.